First, I would like to thank Phi Science and the organizer for inviting me. It's it's a great honor and pleasure to present today the latest work we have been doing at Qatar Genome. So the title of my presentation is the roadmap for genomic research in the Middle East and insights from Qatar Genome program. Yes, so a remarkable milestone in genetics is the 1,000 Genome Project, which sequenced over 2,500 genomes from 26 populations and cataloged over 88 million variants. The resource provided by this project has empowered genetic association studies at large scale and allowed detection of genetic association at low frequency variants. This project marked also the start of effective use of the whole genome sequencing approach, giving, of course, the decline of the price of sequence per genome, which is at an average of about $1,000 US dollar per genome. And among the first population, large-scale population sequencing project is the famous DECODE project in Iceland with over 2,600 genomes sequenced. Another great example of large-scale genome project is the Sardinian project, where they sequenced over 2,100 genomes. Consequently, many countries, well, not so many, let's say a handful of countries have initiated such large-scale genomic project, and I will come back to that in the next few slides. So, however, the next question is, are we covering the full spectrum of genetic diversity in genomic research? In other terms, are we focusing also on populations other than European? Well, the answer is we are still far away, and some population or ancestries are still left behind. As you can see on the left side of this plot, which represents the ancestry distribution of studies in the GWAS catalog database, over 50% of studies published are European studies, followed by Asian studies with around 20%. Comparing to less than 2%, let's say 1.2% studies from Greater Middle East or Native American. This is a huge disparity, and this also true for the individual's distribution in the GWAS catalog. More than 2/3 have European ancestry, while the European represents only 16% of the global population. I think we agree here all that we need to do something about this to balance these numbers and to improve diversity in genomic research. So if you look at this question from a different angle, from a global implementation of genomic medicine or the integration of genomic into healthcare, quite a subject of debate and in this time, we noticed since 2013, the governments of at least 14 countries have invested over $4 billion in establishing national genomic medicine initiatives in order to address the implementation barriers and transitions testing from centers of excellence to mainstream medical practice, so from the bench to the bedside. For example, in countries such as the UK, France, Australia, Saudi Arabia, and Turkey, the workforce and infrastructure development has been coupled with testing large numbers of patients with rare disease and cancer. On the other hand, other countries such as the US, Estonia, Denmark, Japan, and here in Qatar, we have invested in population-based sequencing projects with, when possible, and return of results to participants. There are also other national initiatives such as in the Switzerland, Netherlands, Brazil, and Finland. They are primarily focusing on the development of infrastructure such as common standards and data sharing policies and platform, which is also very important when we come to big data sharing, open science, and collaboration. However, this project will potentially be dominated by the gigantic China precision medicine initiatives. It's a 15 years program, which cost over $9 billion, and which aim to sequence 100 million genomes by 2013. So we need to speed up before the tsunami Chinese come. So Qatar Genome was launched by Qatar Foundation with a comprehensive national strategy based on 7 pillars. Those include integrating with the Qatar Biobank, utilizing local genomics and bioinformatics infrastructure, forging nation research partnership with the local stakeholders and international collaborators, investing in local human capacity, also helping drafting policies and regulations, and building a national genome data network and integration of genomics into clinical settings. And my colleague, Dr. Said Ismail, presented this morning the way how Qatar Genome is facilitating the implementation of precision medicine in Qatar. So QGP first phase was started in September 2015, and its deliverables included the sequencing of over 6,000 whole genome. Also, we launched the QGP Research Consortium. We established a genomic research funding program with Qatar National Research Fund program here in Qatar, QNRF. And the individuals we sequenced in this pilot phase or first phase were participants recruited at the Qatar Biobank. In phase 2, which started last year, we are ramping up our sequencing program and phenotyping efforts up to the mark of around 10,000% of the populations. Hopefully, by the end of this year, we will have up to 25,000 whole genome sequence. The final and the largest phase will start in 2021 up to 2026, and here we are aiming to sequence a large proportion of the Qatari population. So in this phase, we will approach the primary and secondary healthcare centers to secure such large numbers, because biobanks will not be able to provide these large numbers. But please let me know before I move to show some of the first results of the phase 1. I would like quickly to go over some of these pillars of Qatar Genomes. The Qatar Biobank is a national research platform housing extensive lifestyle, physical, clinical, and biological measurements of a large part of the adult population. So the participants are 80 years old, Qatari citizens. The main strength of the Qatar Biobank is that it is a population-based biobank with a longitudinal and prospective wealth of data. This schematic represents the visit workflow of the biobank participants. As you can see, several clinical and imaging data are collected over the time and when the participants visit biobank. In addition to genomics, we are interested also to other omics technologies into medical and research practice. For example, we also do epigenetics, we do transcriptomics, we do metabolomics, and we do proteomics and microbiome for not for all the 6,000 or all the samples, but at least for few thousand of these samples. We also invest in having the state-of-the-art infrastructure at both lab side with several Illumina HiSeq sequencers, and very soon we will be acquiring the new NovaSeq when we start our ramping up our project. At the bioinformatics sites also, we have a powerful high-performance computing platform, and as you can imagine, this is much needing because it's heavy computation and it's a very big amount of data. We are also supported by a strong research network here in Qatar, consisted of research centers and universities like HBK universities with its two research arms, the Qatar Biomedical Research Center and Qatar Computational Research Institute. We have also collaborators, and we work closely with Hamad Medical Corporation. We work closely with Qatar University. So these are the stakeholders here that are supporting our research network. In 2017, we established a research consortium called Qatar Genome Research Consortium, consisting of over 150 local and international researchers working on a predefined project. This is the first project that we started our program with, a project on the GWAS catalog of the Qatari population, a cancer landscape of the Qatari population, a project on the loss of function mutations, another project on pharmacogenomics, a project on the reference population and population structure. And I will talk in the next slide about some of these first results from these projects. So why QGP is unique? I think Dr. Said this morning talked about the uniqueness of Qatar Genome. And so for the sake of time, I will skip this slide. So what we have seen by analyzing the first phase consisted of 2,818 Qatari individuals. So this is in a very brief nutshell some of the numbers. We have identified a total of 88 million variants, among them 74 million single nucleotide variants and 13 million insertion deletions. Interestingly, also, we found 24 million of these variants were novel variants and not previously reported in DB-SNP database or other population database like the GNOMAD-AD or 1000 Genome or the Greater Middle East Project. So the figure on the left, and this the results of this is now under consideration. We have submitted this paper a few weeks ago, and it's now under consideration. So as you can see, the figure on the left shows the comparisons of QGP variants with other population database like DB-SNP and GNOMAD, and the numbers I have said before. And we also evaluated the impact and scale of disease-causing mutation variants in our population, and we annotated the variance list with disease phenotype information from HGMD, ClinVar, and the OMIN database. And in total, we found 4,254 disease-caused mutations variants. The figure on the right represents the functional class of these human gene mutation variants in our data. So next, to capture the genetic diversity of the Qatari population and understand its relationship with the world population in both modern and ancient times, we identified five major ancestries, among them a group of peninsular Arabs forming a unique cluster. And this is an updated results from the previously known results from the Qatari population that were published in 2016, I think, where we found only 3 clusters. So here, with more data, more sample size, we were able to define 5 clusters in the Qatari population. So using this unique data sets, we also built a panel for genotype imputation for the Arabs and Middle Eastern ancestries, which shows an improved imputation score for rare and common variants, for rare and common allele frequency variants observed in the population. So we compared the variants that we found with the variants in 1000 Genome, and using this imputation panel, we see improvement in the imputation score for common and also rare frequency variants. In another paper by colleagues at HBK University and Cornell University, Dr. Omar Al-Berra and Dr. Carsten Sur, we wanted to gain insight into the genetic architecture of health and disease related to quantitative traits. And in paper, which is under consideration now, under review, We have studied 45 quantitative traits, 45 clinically relevant traits, and 6,200 individuals from the Qatari population. And as you can see, these traits and phenotypes range from anthropometric traits like BMI, heights, to some blood cells, counts, enzymes, lipids, and other biochemical variables. And we found several important findings of this comprehensive study, which includes replication of multiple associations reported in the DB-SNP, in Caucasians, but also in Asian GWASs. We described the differences in allele frequencies and LG patterns for replicated loci. And importantly, we also discovered novel genetics associations, mostly with variants common in the Qatari population, but rare in other populations, as you can expect. And as you can see, this is a Manhattan plot, a combined Manhattan plot, showing all the loci that we found by doing this GWAS for 45 traits. We do have more traits, and we will include them in the next paper. We also designed arrays, a chip array, as a collaboration between us, Qatar Genome, Qatar Biobank, Hamad Medical Corporation, Cornell, and Sidra Medicine. And this is the first genotyping array specifically designed for precision medicine in the Middle East, and it is suitable for genetic disease diagnosis, research, and screening. When we started the project in 2015, a year later, we conducted a national survey, and this study is to explore the public attitude of the Qatari population towards genetic testing and towards participating in the Qatar Genome Program. And we have representative samples of 834 adult Qataris were surveyed in May 2016. And the results showed approximately 71% of the participants surveyed reported that they are willing to participate in the activities of Qatar Genome. And the paper is now in press and should be online very soon. So as an integral part of SARS large-scale genomic project is the reporting, and we started also by delivering genomic reports on lifestyle and wellness, including details on food metabolism, response to exercise, and so on. And we will focus in the next phase on pharmacogenomics and polygenic rescoring once we have enough data. Finally, I would like to conclude by the topic of the moment, which is COVID-19. Last week, there are a new sets of results, and Qatar Genome Program contributed to this results by the host genetics initiatives. And please bear with me I want to spend a few minutes introducing these initiatives for those who are not aware of it. So this initiatives called COVID-19 Host Genetics Initiatives brings together the human genetics community to generate, share, and analyze data to learn the genetic determinant of COVID-19 susceptibility, severity, and outcomes. It is led by colleagues from Finland and from the Broad Institute, Andrea Gana and Mark Daly. And the initiatives has three aims. First one is to provide an environment to foster the sharing of resources to facilitate COVID-19 genetic research. Example, we share protocols, questionnaires, IRBs, forms, and to help the community speed the research. We also organize analytical activities across studies to identify genetic determinant of COVID-19 susceptibility and severity. And we provide a platform to share the results from such activities as individual level genotypes, but also as summary statistics, which we will put these results broadly for the scientific community. And last week, the HGI shared publicly the results of the round 3 of the meta-analysis looking at susceptibility and severity of COVID-19. And we are proud, as you can see in the list on the right side corner, that Qatar is the first contributor from the Arab world in this initiatives, and we used our retrospective data of 15,000 participants so far to analyze to perform analysis COVID tested positive versus population. So the results of the round 3 are in the slides, and we found a strong signal at chromosome 3, and this signal is quite robust and consistent across the participating studies. It is a spanning cluster of a gene SLC-A20, CCR9, CXCR6. Several of these genes have functions that are potentially relevant to COVID-19, like this gene called SLC6A20, which functionally interacts with AC2 gene, the famous receptor for SARS-CoV-2. As you can see in the forest plot, the results of the Qatari analysis are quite consistent and aligned with the results from other large cohorts like UK Biobank, Genomics, England's Lifeline, or Netherlands Twin Register. We are also participating in the national efforts here in Qatar to sequence the viral genome, and we work with our colleagues at Qatar University to sequence the viral genomes using the Oxford nanopore technology. So by this, I come to the end of my talk, and I would like to thank the teams at Qatar Genome, Qatar Biobank, as well as our collaborator at Hamad Medical Corporation, CIDRA, the HBKU, and other institutions, and I thank also the participants in this study. Thank you for listening, and I'm happy to take any question.